We're going to take a look at aerobic respiration and fermentation. So just a reminder where we're going to be looking at in the cell. So we're going to be looking at these catabolic reactions and focusing mostly on the mitochondria and making ATP. Aerobic respiration is also called cellular respiration. Here's the chemical reaction. We're going to start with our two reactants. We've got glucose and oxygen. And the glucose we're going to break apart into carbon dioxide. And then the oxygen will get turned into water. And we're going to break all the chemical bonds in the glucose here. So if I represent the carbon atoms as little balls, and we break them apart into the 6 carbon dioxides, you can see the 6 carbons here have all been separated. We're going to release all the energy stored in the chemical bonds there and then temporarily store that energy as ATP. And we're going to generate up to 38 ATP per glucose molecule. So is this going to be an anabolic or a catabolic reaction? So hopefully you said this is a catabolic reaction because we're taking a larger molecule and breaking it apart into smaller molecules. And is this an endergonic or exergonic reaction? And this is an exergonic reaction because we're releasing energy. And then which reactant is oxidized and which reactant is reduced? So our two reactants to choose from, we've got glucose and oxygen. So the glucose ends up as carbon dioxide on the other side. So you can see that it loses hydrogen atoms or electrons. And remember, oil, oxidation is loss of hydrogen atoms or electrons. So glucose would be oxidized, that's the oil part. And then oxygen turns into water, and you can see that the oxygen gains hydrogen atoms or electrons. So that's the rig part. So oxygen will be reduced because it's gained hydrogen atoms or electrons. So to fill in our answers here, we'll have glucose will be oxidized, and then oxygen will be reduced. So there's 4 stages to aerobic respiration. So I just want you to know basically what's happening in each of the 4 stages and then where it's occurring in the cell. So glycolysis occurs in the cytoplasm, and then the other 3 steps all occur in the mitochondria. So here's a picture just showing the mitochondria over here, and then we've got the cytoplasm out here where glycolysis occurs, and then forming the acetyl-CoA, the Krebs cycle or citric acid cycle, and then oxidative phosphorylation. These 3 steps will all occur within the mitochondria. So basically what's happening in glycolysis is we're going to take glucose, and if you notice the name glycolysis, lysis means to split or break apart. So we're just going to take the glucose and split it in half into 2 pyruvate. So I just want you to know glycolysis, we're just splitting the glucose into 2 pyruvate, and that occurs in the cytoplasm. And then step 2 of aerobic respiration, we're going to be forming acetyl-CoA. This occurs in the mitochondria. So we'll just take the pyruvate and then we'll just make that into acetyl-CoA. And then as we do that, we're going to chop off one of the carbons, and that becomes carbon dioxide, which is a waste product, which we'll exhale. Step 3 is the Krebs cycle. So this occurs in the mitochondria. And we're going to do 2 turns of the Krebs cycle because we have 2 acetyl-CoAs. So we're going to feed one through each turn of the cycle. And we end up making some ATP. And then we've also got these electron carriers. So we've got NADH and FADH2. And then we've got waste product carbon dioxide, which we'll exhale. And then our last step, oxidative phosphorylation, also occurs in the mitochondria. And what we're going to do in this last step is we're going to take the electrons that are in our electron carriers, NADH and FADH2, and we're going to drop them off in the electron transport chain. And they're going to go down the electron transport chain and release their energy, and that energy will be used to make ATP. And then at the end of the electron transport chain, we're going to give the electrons to oxygen. And once oxygen accepts those electrons at the end of the electron transport chain, we make water. So oxygen is required for aerobic respiration because oxygen is the final electron acceptor. And that's at the end of the electron transport chain. And ETC is just electron transport chain. Okay, and then this is just a close-up, just showing you the inside of the mitochondria. Remember, there's a double membrane. There's an outer membrane and then the inner membrane, and the inner membrane is all folded up into these cristae, which increases the surface area. And in this inner membrane of the mitochondria, embedded in there is the electron transport chain. So that's where the NADH and FADH2 will drop off their electrons, goes down the electron transport chain, and at the end of the electron transport chain, the electrons will get accepted by the oxygen, and then that makes the water. And then also in the mitochondrial cristae in the inner membrane, we have ATP synthase, and hydrogen ions will go down through there, make the ATP synthase spin, and that'll provide the energy to make the ATP. So to answer our question, where are the electron transport chain and ATP synthase located in the mitochondria? That'll be in the mitochondrial cristae, or the inner mitochondrial membrane. And then this is just another close-up, just looking at the inner mitochondrial membrane with the electron transport chain and ATP synthase. And then here's the outer mitochondrial membrane. So just a summary of aerobic respiration. 1 glucose is going to give us 38 ATP. If no oxygen is available, the other way that we can make ATP is through fermentation. And the electron transport chain in the mitochondria will not be used in fermentation, so only glycolysis will be used. And in glycolysis, we're only able to make 2 ATP per glucose molecule, so it's not very efficient. Whereas in aerobic respiration, we're making 38 ATP per glucose. Here with fermentation, we only make 2 ATP per glucose. And what we're going to be doing is we're going to be taking the electrons at our electron carrier, NADH, and we're going to give them to an organic molecule like pyruvate to regenerate the NAD+, and we end up making lactic acid. So here's just a picture of that if we do have oxygen present, we'll go ahead and use our mitochondria because we can make a lot more ATP per glucose that way. But if oxygen is not available, we can't use our mitochondria, then we'll do fermentation. We'll just use glycolysis out here in the cytoplasm, and we end up making lactic acid. So here's what happens in fermentation. We're going to take our glucose, just do glycolysis. We'll split it in half, make our 2-pyruvate. We make only 2 ATP per glucose. And when we do glycolysis, we're going to take some electrons off the glucose, put it onto our electron carrier, make NADH. And the way I think of NADH is like a little dump truck. And it carries electrons. So here's our NADH. So it's picked up those electrons from glucose. And normally NADH, if oxygen were available, it would take the electrons to electron transport chain in the mitochondria and dump them off there. And then it can come back and get more electrons from the next glucose and keep going. But we don't have our mitochondria working because we don't have any oxygen, so we need some place to dump those electrons off so we can empty the dump truck and then go back and pick up some more electrons from the next glucose, and then we can make another 2 ATP. So we're going to take those electrons on NADH and dump them onto pyruvate, and that's how we make lactic acid. And then we've got our empty dump truck, or NAD+, ready to go for the next glucose, and then we can make another 2 ATP and we can keep going with fermentation.